My name still remains Adeola Michael. I am here today once again to continue our discussion in genetics, and that will be in furtherance of the thematic area of Omega Bio-Edutainment. Last week, we in one of my earlier video in genetics, we were able to look at pedigree, application of pedigree, and consanguinity. And in dealing with that, we were able to see that consanguinity as well as a pedigree, these are tools that they use in genetic counseling in order to be able to diagnose and predict the inheritance pattern of some traits. And in looking at that, I was able to tell us how bad blood are being inherited within the family. And intentionally, I use that word bad because my focus was actually to deal with some of the genetical diseases. And we were able to look at 4 categories of genetical diseases that we can actually use pedigree to be able to diagnose and also predict what might be the outcome in the next 1, 2, 3 generations. And we have been able to actually use this symbol, that is pedigree symbol, to be able to peep into what the inheritance pattern is actually going to be. Now, I want to classify that into 4, just like what we did in the previous video. I classified them. I made mention of case studies of um, pedigree, using pedigree in order to be able to know exactly what is going on. Now, the first one was the case study one was on autosomosis, and which was another autosomal recessive inheritance. And we were able to look at the inheritance pattern of that to be 25, 50, and 25. That is, those that will be affected, um, they have the chance of being affected to be they have 25% of being affected, 50% chance of being a carrier, and 25% of being unaffected. We look into this, and also X-linked recessive inheritance. We also look into the case of hemophilia, and we also look at the mitochondrial pattern of inheritance, whereby we look at Leber's hereditary optic neuropathy. And just to complete that aspect, I just want to mention what are the application of pedigree. I want to actually mention this behind the camera because it was part of what we're supposed to have done. So the first application of pedigree notation is the use of it in genetic counseling, just like we have been able to say. It is used in genetic counseling. Then number 2, it is also used in tracking genetic diseases. If we want to track genetic diseases, it is something that we can use. Then number 3 is is used in public health. It is used in public health. So, and public health and research. So we need to also understand that. Then the fourth one, the fourth application of it is that apart from using it in genetic counseling, we can also use it in what is called personalized medicine. So based on pedigree, we can actually tailor some preventive measures like early screening and treatment option for genetic counseling. So without wasting our time, today we are going to continue our discussion in genetics on a new note, and that is going to be introduction Introduction to genetic disorder. Introduction to genetic disorder. So I'm going to start this on the ground that why are we so interested in this? What is this? So the first thing is, what do we mean if you are actually talking about genetic disorder? You need to know that genetic disorder in human, this is basically in human, no, in human. So genetic disorders in human are diseases that are caused by changes in the DNA sequences. So definition, okay, genetic disorder in human, these are diseases that are caused by changes in the genetic makeup. Okay, so diseases due to changes in DNA sequences. Don't forget that whatever affects the DNA might eventually going to affect the gene, and you know what the gene do in the body. Okay, so that will tell you that that kind of abnormality can be carried from parent to the offspring. Okay, that affects the structure. It could be that affect the structure and or function of certain genes. Don't forget what genes are. Genes are segments of DNA. So, and whatever affects the DNA might and is going to affect the gene at a point in time, and this effect is going to be might affect the structure and the functionality of that gene. We are going to see some basic examples of this. So after the definition, the second thing that we want to look at is the examples. We want to look at examples. Okay, let's say types of genetic disorders in human. The first category or classification is what we can classify as single gene problem, which is termed as monogenic. That is, if the problem is just within one gene, it is called monogenic genetic disorder. Okay, and it could also be so it can be multifactorial, from the word multifactorial disorder. Okay, then it can be chromosomal. That is, when the any of the chromosomes is affected. Any of the chromosome is affected in human. Don't forget that there are 23 pairs of chromosome, which I said the first 1 to 22 are called the autosomes, and the other one that is also is called the sex chromosome. So the pattern of disorder. Okay, this one is multifactorial disorder, chromosomal disorder. So anyone that now has to do with affecting the chromosome, it is called chromosomal disorder. Then the fourth one is mitochondria. mitochondrial dysfunctioning or disorder. Mitochondrial disorder. So it could be any of these four. So if you now to classify the genetic disorders, it can be classified as a single gene, which is called monogenic, or it can be multifactorial, it can be chromosomal, as well as it can also be as a result of dysfunctioning of a mitochondria. So after that, we now want to look at the reason why are we actually interested in this. So that's going to lead us to what are the objectives of this, or what is going to be the take home at the end of this lecture. What should we take home? What should be expected of us? Now, the first one is that it is expected that to know and understand, to know and understand the what is called genetic disorders. That is, what are they called? That is definition of that. Then to know the types. Not only the types, we want to be familiar with other basic concepts. So we are going to familiarize ourselves to other basic concepts of in genetic disorder. Then the second thing is to be able to understand. We should be able to understand the mechanism, that is how the mechanism of inheritance, the mechanism of its inheritance pattern. Okay, then the third one is that we want to be able to discuss, the medical, want to look at the medical implication. Okay, not only that, we want to know, because this one has to do with to understand the mechanisms of inheritance. This has to do with the diagnosis, that is how can it be diagnosed. So this next one is to be able to discuss the medical implication, psychological implication, as well as the social impact. What could be the social impact of each of the genetic disorders? Could it be, if it is a single gene, which is monogenic, multifactorial, chromosomal, or mitochondria? So we want to know what might be the psychological, the clinical implication, and the social impact of this. Then the last one is going to be, we want to be able to review the current trend, to review the current trend or the options, treatment options that are available, current options, and be able to advocate, because the essence of being a genetic counselor is to be able to be an advocate. Advocate that bad gene should be left out. That is, we should be able to do away with bad things, and we should be able to look at and promote what is going to lead to propagation of the good gene. And that is by implication, that is exactly what is called what's the essence of genetic counseling. The essence of genetic counseling. We are going to see most of this as we actually go on. So advocate effective therapy We want to look at the effective therapy. And effective therapy, okay, sorry. Let's say this one is going to be comma. So, and is going to come here and be able to recommend. We should be able to prescribe. Let us use the word prescribe. We should be able to prescribe the possibilities, if possible, the possibilities of phasing out let's use phasing like this, phasing out the bad genes. Okay, so that is the whole essence of this topic. The first one is we want to introduce us into understanding what genetic disorders are, and what are the types of genetic disorders, as well as we will also acquaint ourselves to the basic concepts in genetic disorder. Then the second objective is to be able to understand what mechanism, the mechanism of inheritance. That is, we want to be able to diagnose, and part of the tools that we can use in diagnosing it is what is called the pedigree, what we have actually used. So that is to understand the mechanism of inheritance. Then we should be able to profile or discuss the medical implication. Should be able to profile, we'll be able to discuss the medical implication, clinical manifestation, psychological impact, as well as the social impact of a genetic disorder. Then the last one is to review the current available options. We should be able to review the current. We will review it one after the other. Then, if we now have, then we should be able to advocate. That is, we have to be an advocate of effective therapy, and possibly we should be able to prescribe the possibilities of phasing out the bad genes in our population. And this field of phasing out bad gene is another thing entirely in molecular biology, or in what is we can call genetic engineering or counseling. It is called eugenics. So don't forget from your elementary understanding of eu, that is a true or good. Genetics, that is anything that has to do with genetics, or anything that has to do with gene. So the practice of promoting promoting good gene at the expense of bad genes is called eugenics or eugenics. It is very, very possible. So, and the essence of genetic counseling is that so that when you are able to diagnose what might be the problem, you now be able to promote the problem. You will be able to prescribe the way out and be able to promote the good one, and that is the practice of eugenics. So we are going to see all this as we go on. So what we will just do is just to start one by one. We'll start picking this one after the other. Don't forget that I told you that our concern today is on genetic disorder in human. And if we are talking of genetic disorder in human, what do we actually mean by genetic disorder? And I said that these diseases that are due to changes in DNA sequences that can affect the structure as well as the function of any gene in human is called genetic disorder. So it is also what is also called we also come across it as a genetic. Some people will call it genetic syndrome. So and we have been able to look at different types of disorder. The first one is a single gene disorder. That is when there is problem in only one gene, which is called monogenic. Then the second one is a multifactorial. Multi from the word many. That is as a result of combination of problems coming together and manifesting is called multifactorial. There are different factors that can actually cause that, including spontaneous, induced disorders, as well as others. So we are going to see all this as we proceed. The third one is a chromosomal disorder. If any disorder is as a result of the manifestation or that has problem that has the origin from the chromosome, and that is what I called in my earlier lecture, I called it bad gene or bad blood. That is a chromosomal disorder. And I said, as a kind of reminder, that there are 23 chromosomes in human. The first one to 22 is called autosomal, and the 23rd one is called the sex gene, which actually determines whether we are going to be boy or we are going to be a girl. So the gene that is responsible for femaleness is an XY, will manifest itself as XS, and the one that will be responsible for maleness, it is called XY. So we saw this in the earlier lecture. So then today, the last one is a mitochondrial disorder. We have been able to diagnose this using pedigree. So, and just like I said, the question is, why are we so interested in this kind of topic? Because there must be a reason. At the end of this lecture, at the end of our consideration of this lecture, we should be able to have understood. We must have been able to get ourselves exposed to the basic concepts, the different types of genetic disorder, and we should be able to have known what genetic disorder are. What are they? That is the definition. That is the objective one. The objective two is that we actually want to understand what might be the mechanism of inheritance. That is, what might be the pattern of inheritance. How is it going to be inherited? Is it going to be inherited as autosomal recessive, or is going to be dominant? So we want to look at this pattern of inheritance, and that was one of the application of what is called pedigree. And don't forget that pedigree is nothing but symbolic representation of traits or genetic diseases that will be able to use for prediction or pattern of inheritance among any given population. So the third one is that we actually suppose we want to know if we are going to to be able to discuss the genetic implication as well as the sociological implication and social impact of this genetic disorder. We want to know that. Then the next one, the last one, is that we want to review what the current options are that are available to us. We want to review those current options, look at the current trends, look at the events that is actually surrounding or affecting the issue of genetic disorder. Then we should now be able to advocate for effective therapy, as well to be able to see if we can start prescribing measures that could lead to phasing out the bad genes. So, and I call this one the practice of phasing out bad genes and encouraging the good one. I call it eugenics. So we are going to see all this as we go on. So now I want to start picking them one after the other so that we'll be able to understand. We should be able to understand them as we go on. So don't forget that we are still considering genetic disorder in human. Genetic disorder in human. Okay. So so we will start picking them one after the other now. And the first one we will look at, the first one, okay, before we go into that, in this topic, we are going to let's try and identify the subcontents. Okay, let's identify subcontents under this. The first one, just like I told you, the first one we are going to consider as okay. So we might be able to conclude today, and we might not be able to conclude in this video. But what the first thing that we want to look at, which is a subcontent, is that we want to look at genetic causes. What are the genetic basis or the genetic causes of genetic disorder? What are that? So under that, we want to look at types of mutation. We'll look at type of mutation. Intentionally, I won't be able to go into the chemical basis and the genetic basis by considering examples or case studies at the genetic level. And the reason is just that this will be dealt with in our future lessons. So we are going to look at this into details. Then types of point mutation. After that, we will look at genetic inheritance pattern. Genetic inheritance patterns. Okay, we'll look at them. After that, we now look, the second thing, broad topic we will look at is what are the clinical, I told us as part of our objective, what are the clinical manifestations of genetic disorders. Okay, how do they manifest genetically? Anybody that is having any of the genetic disorders, what are those things that you will see? So that the doctor will be able to understand what the symptoms could be. Okay, so under that, we will look at common clinical features that you can see, which is general. So then we'll look at the common clinical features. Then under that, we'll consider examples. We'll look at case studies, examples of genetic. We'll pick them one after the other. Genetic disorders, okay, and their manifestation. and manifestation. Okay, this is very very important. So after that, which is the third, we want to look at the diagnosis 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 of genetic disorders. So the practice of the application of this genetic counseling will actually help you to know who should you marry, who should you marry. So it's not we do not actually believe in scientifically there's nothing like for better for worse, because just like I used to say, there are some people that they can never be a father, they can never father any child. And if somebody that is genetically matured, or that is genetically favored to give birth to as many children as possible, if they now marry each other, you know that at the end of the day, that kind of marriage, there's nothing like for better for worse in that. So before you actually go into that marriage, it is better you seek what is called genetic counseling. Genetic counseling, and this is part of that's going to be the concluding part of this topic, because at the end of the day we'll look at what do we call genetic counseling, and what are the best practices of genetic counseling, what are those inheritance, or what are those diseases, what are the probable advice, or what are the scope of issues that can be brought for genetic counseling. We are going to see this in the long run. But don't forget, one of the reasons of this is to be able to know genetic disorders, be able to know their manifestation, so that you won't actually make a wrong choice sometimes. One of the reasons of seeking genetic counseling is not to make a wrong choice. So under the diagnosis, we are going to look at what is called genetic testing. What tests can you do or can be prescribed, so to be able to, that is genetic testing. What are the genetic testing to know whether the problem is as a result of point mutation, is a result of insertion, is as a result of deletion, or the kind of genetic genetic eye that we can use in diagnosing this. Then we will now look at what is called karyotyping. Karyotyping. So karyotyping has to do karyo, from karyo, that is a chromosome. So we have to look at different types of chromosomes, different chromosomal disorder or chromosomal aberrations, and then we'll be able to see which of these diseases, how and where it can be actually diagnosed. Then we'll now look at what is called prenatal screening. Sorry, prenatal, that is before birth, prenatal screening. So even before you prenatal, that is pre, from before, prenatal screening, we look at that, still under that. Then the fourth thing we are going to, you see, is going to be a very long lecture series on that, but to me it is very very interesting, because it is what we're supposed to know. Okay, so the next one is we'll look at available options. We'll look at treatments of genetic disorders. Is it going to be when the genes is bad, or when the genes are bad? Is that going to be the end of it all? We are going to look at it. So under that, we are going to look at what are the general treatments that could be prescribed. What are the general treatments uh, that are available. So we look at general treatment approaches. Okay, so this will include symptomatic treatment, gene therapy, prenatal gene editing as well. We are going to see all this. In fact, more. I'm very sure that more classes will also join us in this area. So then the second thing we will look at is that what are the specific treatments? What are specific treatments for certain selected disorders? For selected disorders, okay, we are going to look at what are the available treatments, which could be a kind of general treatment approaches, or those ones that are this. Then we now cap it all with the fifth point, which is going to be prevention. Okay, and the role played by genetic counseling. Genetic what? Counseling. Okay, so you can see that we have a very long way to go. Because we are going to treat this topic under 5 broad subtopics. The first one is genetic causes. We'll look at point mutation as well as a genetic inheritance pattern. Then we are going to look at clinical manifestation of genetic disorder, which include common clinical features, example of genetic disorders and their manifestation. So, but whichever one that is point to note, there's very big points that I want you to note here. Very, very big points. So, genetic disorder, although they have kind of genetic basis, but they are of prime importance. They are of very prime importance, not only to us as geneticists or biologists. It is very prime importance in medicine, or what is called medical genetics. Very, very important. It is very, very important in maintaining social bond, marital bond. So marital bond, as well as it has psychological. It also has a psychological importance. So very, very important. In fact, to me, I think we ought to play. We ought all to get involved as an advocate of what is called what? Genetic counseling. That is prenatal screening. It is very, very possible and very, very important that all of us, we should begin to change our orientation, especially before we say yes forever. So don't forget that genetically speaking, there are nobody there's nothing that is identical. Nothing like identical twins. Nothing like identical, because even what we call the monozygotic twins that are called identical, we have certain differences, millions variations. We have variations that will prove to us that normally, as a geneticist, there's nothing called identical. Nothing called identical twins or monozygotism in genetics, because at the end of the day, the fingerprinting pattern is not going to be the same. Not only that, there are different changes, different types of changes at the genetic level. So we need to know this. Now, because of the long drawn subtopics, I think what we will be able to do today is just going to be that of number one. But let me try and display uh, what we have on the screen so that you'll be able to take them down. Okay, so don't forget that we said the first thing is different subtopics. Different subtopics that you have See the difference of topics that we have under genetic disorder. The first one has to do with genetic causes of genetic diseases, genetic causes of genetic disorder, and we will look at types of mutations that are actually involved, the type of mutation that are involved. So, apart from that, we will look at genetic inheritance pattern, clinical manifestation, and so on and so forth. I'm very sure that you are able to copy what we have on the screen. The second one is a clinical manifestation of genetic disorders, which include common clinical features, as well as we look at examples of genetic disorders, examples and their manifestation. The third one is we are going to look at diagnosis of genetic disorders in the area of genetic testing. We'll do something we call karyotyping. Then we are going to get ourselves involved in what do we understand and what is the role of prenatal screening, that is before birth, that is even before marriage. What can we do? So then the fourth one has to do with prevention of genetic counseling. Treatment of genetic counseling. Treatment of genetic counseling. We'll look at general treatment approaches. Then we'll look at specific treatment for selected disorders. So after that, we'll now look at prevention and genetic counseling. So, and I want you to know that genetic disorder, whichever point or whichever perspective that you want to look at it, it is of medical importance. Medical importance. It has to do with maintaining or disrupting social bond. It's either break or mar it, marital bond, as well as it has psychological importance. And because at the end of the day, what's going to be the overall implication or application of knowing things about genetic disorder will be to have a very robust society, a society that is actually free of bad genes, and that is what is called eugenics. So, society that is free of bad genes. So, don't forget, that is telling you that if we talk about bad genes, that will include a lot of things, including measure of intelligence. Okay, so we are going to see all this as we actually go on. So just like I told us that we might not be able to go further than just picking the first point, which has to do with genetic causes of genetic disorder. What are the genetic causes of genetic disorder? So we are going to look at them. Okay, so and that will be where we are going to stop today, before we now try and devote 2 or more videos for this. Okay, so the first one is that we want to look at genetic disorder from the perspective of, okay, so we want to look at genetic causes of Genetic causes of genetic disorder. Okay. So, and the first thing we look at is what is called point mutation. Point mutation. Don't forget that mutation is a sudden change in genetic makeup. So, I just like I said that we won't be able to go into the details of how to be able to illustrate it with what will happen at nucleotide level, how the nucleotide level will look like if it is undergoing point mutation. We won't be able to go into details of it, but we will go more into it maybe in the next lecture. In fact, we have to look at the chemical and the chemical manifestation or chemical mechanism of point mutation. We'll look at mutation generally and we'll look at what is going to happen. But at this level, we just want to look at types of mutation. So we have point mutation. So point mutation, this is telling you that just a single nucleotide is altered, is affected. A single nucleotide is affected. So we will look at this. An example of this, we have e.g. sickle cell anemia. Just an alteration in one. Okay, sickle cell anemia. Okay, sickle cell anemia. So, and this kind of mutation, this point mutation could be point mutation, that is when a single nucleotide is being formed, it could be in form of insertion. Insertion has to do with when only one single nucleotide is added. Okay, it could be in form of deletion, or when a single nucleotide is deleted. Okay, sorry, this is a different thing entirely. So a case study of sickle cell anemia, for example, this kind of a replacement single nucleotide can result into a protein, different types of point mutation, which can be mean-sense mutation. It can be a silent mutation, or it can be a nonsense mutation. So we will see what do we mean by mean-sense, what do we mean by silent, and what do we mean by nonsense kind of mutation. So the second type of point mutation is mutation that has to do with either insertion or deletion. In some other places, you can also receive them as indels, short form, indels. Okay, insertion and deletion. That's why they call it indels. That is when extra nucleotide chromosome is being added or deleted. So this can cause what is called a frame shift. It can result into what is called frame shift mutation. Frame shift. I won't be able to go into the detail because this is not molecular class anyway. But don't forget that protein synthesis actually takes place in the ribosomes, and the process is called translation. So it's going to involve transfer RNA, ribosomal RNA, and messenger RNA. So it is the messenger RNA that will line up series of codon that will be interpreted in triplets. And on the surface of the transfer RNA, there are anticodons. That is going to be complementary to the codon that will be on the messenger RNA. And only two transfer RNA is going to be what? Is going to occupy the ribosomal RNA at once. So if there's now insertion or deletion that is as a result of that will result in what is called frame shift, that means that the transfer RNA will now jump. We jump over. We jump over. That is why they call it a frame shift. We are going to see case study of this as we actually go on. And the case of this, is a condition that is known as Duchenne syndrome. Duchenne muscular dystrophy. So we'll look at this as we go on. Okay. So the third type of point mutation, or the point type of genetic cause of genetic disorder is that the first one is a genetic point mutation, which could be as a result of a single nucleotide is just affected in the case of sickle cell anemia, and it could be nonsense, it could be silent, or it could be mean sense. So, and when we now have a kind of addition or deletion, which will cause a kind of frame shift. So if it is insertion, that means extra nucleotide will now be involved. And since nucleotides are being that code for amino acid will always be in triplet, so there will be a kind of frame shift. So example of that is what is called a Duchenne muscular dystrophy. Okay, so then genetic disorder could be as a result of what is called duplication. It could be as a result of duplication. Okay, and that means that there's going to be a repeat of certain segments of the DNA. So a repeat of certain segments of the certain part of a DNA. An example of that is what is seen in this condition. People in this condition, Charcot's Marie Tooth disease. We look at a clinical manifestation of all this by the time we get to point 2. We'll start picking them, all these conditions, we'll start picking them one after the other, and we'll look at what will be the clinical manifestation of each. Then the last cause of a genetic disorder could be as a result of infarction. Okay, or infarction, or what is called, it could be as a result of a translocation. So this is a kind of rearrangement. In fact, this is going to be a kind of rearrangement. Okay, rearrangement of chromosomes. Rearrangement of chromosome that can disrupt the functionality, that can disrupt the functionality, okay, the functionality, a sample of what happens in hemophilia. Okay, hemophilia, which is a hemophilia. Okay, and certain cancers, which is as a result of translocation of certain cancers. So this is exactly four different types of causes of genetic disorder to be as a result of a point mutation, which has to do with a change or an alteration in a single nucleotide. So, and this one is seen in people with sickle cell anemia, and it could be mean sense, it could be silent, or it could be nonsense. So then the second cause could be as a result of insertion or deletion, which is short form is called indel, which will cause a frame shift. An example of that is Duchenne muscular dystrophy. Okay, then it could be as a result of duplication, which is manifesting in a condition of Charcot-Marie Tooth disease. Okay, then it could also be as a result of inversion or translocation, which is due to rearrangement of chromosome, which can alter or disrupt the function or the functionality of the genes. Or okay, example of that is hemophilia and certain types of cancer. So just like I said, that's going to be the only first aspect that we are going to do in this lecture series. So we are going to continue. So we are still going to continue to look at how each of these is going to look like at a molecular level. So we'll start picking them one after the other, and we'll pick a case study in each of them, and we'll be able to see how it is going to be. So don't forget that this is part of the educational series of Omega Bio-Edutainment. And just like I said, and you know that after each lecture, you are going to expect that you should attempt, uh, you should be able to attempt some multiple choice objective questions under that. So immediately after this video, I'm going to deposit the link that you will use in answering question, multiple choice question between 20 and 30 questions before you will be able to move into the second and other lesson. So I want to really appreciate you for listening. Thank you.